using zooarchaeological data and modern genetic data. And over the last couple of years, also a lot of aDNA studies have contributed to reconstruct this process. Now, one center of pig domestication was the fertile Crescent, and the first domestic pigs appeared around 10,000 years ago there. However, in my talk, I will focus only on northern Germany. And as you can see here, northern Germany here, we were at the far receiving end of the Neolithic expansion. So whatever we received it very late. So the question is, did we also receive the domestic pigs from the Near East, or were the pigs locally domesticated up in the north? Both scenarios are possible. Also the local domestication, because we've always had large wild boar populations there and still have them. Okay. Apart from the question of origin, there are other questions that have been addressed concerning Sus domestication. One question is, how long did the whole process happen, and how did it happen? What were the genetic changes underlying the early domestication process, and which phenotypes were selected on early? The first answers were provided by Gregor Larsen and his team. He performed an ancient DNA study using mitochondrial DNA, in particular an 80 base pair segment in the control region. And the first step they did, they analyzed the haplotypes in present-day wild boar populations, and they identified 4 different haplotypes. And he called them A, C, Y1, and Y2. Now, A and C are only one mutation removed from each other, just as the 2 Y haplotypes. However, Y and the C haplotype, there are 3 steps removed. And when they looked at the phylogeographic, after a phylogeographic analysis, they realized that there is a particular distribution, very clustering. So we find the red ones, the A and C, only in Europe. So these are the European types, and I will refer to them as Eurotypes. Whereas the yellow ones, Y1 and Y2, are located or are restricted to the Near East. So these are the Eastern types. And then Gregor and his team, they performed an ancient DNA study. So first they looked at ancient wild boar, and they saw that the few specimens they looked at, they all carried the Eurotypes, which was expected. And then they selected samples from across Europe, a wide area in Europe, and also a wide time frame. And they saw that in the early Neolithic, they saw in the domestic pigs both the Eurotypes and the Near Eastern types. Mainly the Y types, the Y1 type, more common than the Y2 type. And they suggested that the Y1 type might have been spread along the rivers, maybe the Y2 types across the Mediterranean. Now, interestingly, at later periods, the Near Eastern types disappeared completely, and they only saw the Eurotypes. And also in modern European breeds, we only see the Eurotype. The only exceptions are breeds that we know derived from crossbreeding with Asian varieties, sort of in the last 200 years. Okay, so Gregor concluded that the domestic Near Eastern pigs were introduced into Europe by the early farmers, that later on local wild boar were incorporated in the European domestic stocks, and that the Near Eastern types, haplotypes, were rapidly replaced by the Eurotypes. He estimated that it took only 500 years, which is fairly rapidly. Now that's where we started, where we came in and we asked questions, well, how did this incorporation happen? What type of animals were incorporated? We also asked, well, was it really that fast, this replacement? And we also asked, why were these lineages replaced? Was it just drift, possible? Or maybe there was selection at play. Anyway, so we In the following, I will present you our approach, and I can give you some preliminary data. So our question was, we wanted to determine the origin of the empty haplotypes in northern domestic pigs in northern Germany. We did some mathematical modeling. So the database, our database consisted of 116 ancient Sus remains, and we also used the same segment that Gregor used before, the 80 base per mtDNA segment. We selected 28 wild boar remains and 88 domestic pigs. They were classified only based on biometrics, so basically only based on size. They're both from northern Germany, which means that they were north of the river Elbe, and also southern Germany. And we looked at a period of from 5,500 to 3,000 BC. These are the haplotypes we established, a total of 116. And you see that there is already a very different haplotype distribution in the north and the south. So we considered the following scenario. We considered the time frame of the very early Neolithic in northern Germany, which was, happened more or less overnight. And we looked at the domestic pigs in the north and asked ourselves, well, where did the mtDNA lineages come from? One possibility was they came from the south, because in the south, the domestic pigs had been introduced much earlier. Or the alternative is they did already their own business and domesticated local wild boar. For the modeling, we took into consideration the haplotype distribution using the maximum likelihood estimator. That's a common approach. And we used the data from the preceding period. And we asked, well, what's the probability that the mtDNA haplotypes in the domestic pigs in the north derived from the wild boar in the north? And the answer was 0, which means then that all of the lineages came from domestic pigs from the south. Well, that was not really a big surprise. That's what we expected, because that's consistent with the model that Gregor had already established. Then we looked at the next time slice, so to speak. Here we looked at the middle to late Neolithic period in northern Germany. And again, there were two scenarios. The haplotypes derived from the south, from domestic animals from the south, or from the north. But in the north, it's a bit more complicated now, because at that time they had already their own domestic pigs and the wild ones. So first we asked again, well, what is the probability that the haplotypes derived from the north? And here it was something between 90 and 100%, which means that the majority of the animals came from the north, so the transport or the spread from the south had decreased. Okay, so far so good. And so which animals did they then use for the breeding studies? Was it the wild ones or the domestic ones? So we looked at the probability that they derived from the domestic northern pigs. And this was really a surprise, because it revealed something like between 0 and 20%. Oops. This means that actually the large majority of the animals were wild animals that were crossbred. So what we can summarize from our study is that, consistent with Gregor's model, that there was an influx of Near Eastern female animals from the south at the very beginning of the Neolithic. So, but later on, local female Eurotype animals were bred in, and to our surprise, it was a large extent of wild animals. Of course, we don't know if they also used females, also used male animals. This we don't know. We haven't looked at the Y chromosome distribution as yet. But what has transpired is that they bred, or that they had or managed a hybrid population for hundreds of years by continuously crossing in at least female animals. And the question immediately that we have, why did they do that? That's a lot of work. I mean, and our suggestion is that maybe they had a very small founder population. Remember, northern Germany is, was at the very receiving end. So maybe not many animals made it up to the north. And this population, this fauna population, domestic fauna population, was very precious. Maybe consisted of only relatively few animals. And the problem then that arose was that this fauna population wasn't so stable. And in order to stabilize it, they crossed in wild animals. And we know from modern studies that hybrid populations are very resilient and very resistant also to diseases. At the same time, though, they started to select for domestic traits. We see this, for instance, in the size reduction. And also on this historical painting, you see a female wild boar, and these are her piglets. There was hybridization going on with a male domestic pig, and you see how they differ only in coat color, for instance. And so it must have been quite easy for the early farmers also to pick out those piglets that they wanted, and that had those traits that they really wanted to keep, such as, for instance, coat color or size or behavior. And this brings me to the second part of this talk, which is change in phenotype. Yeah, humans put a lot of selective pressure on the first populations, and we know from studies of other animals that coat color changes were the first that occurred. But also other changes like skeletal changes, muscle growth, fat deposition, fertility or behavior. So we thought about an ancient DNA phenotype study, and one advantage is that based on various modern pig genetic studies, we have lots of data for these breeds, and we also have their associated domestic phenotypes. And also a few genomes have been published in the meantime. Now, what was needed? Well, we said the first approach was not to sequence the whole genome, but rather to focus on certain phenotypically relevant genes or markers, then to enrich them. And as a reference, we wanted not a modern wild boar, but we went directly for the ancient wild boar. And of course, we wanted to look at various ancient domestic pigs from different periods and different regions in order to reconstruct the phenotype. Now, I'm coming from human genetics, and phenotype reconstruction in humans is very difficult and very frustrating. Now, so it's a nice change to work on domestic animals, to be honest. But also in animals, phenotypes are usually polygenic and even worse, multifactorial with a lot of environmental factors. But in domestic animals, fortunately, we have usually a very large effect size. And for instance, just one QTL can explain like between 15 to 30% of the phenotypic variation. This is wonderful. This is something we would dream of in human genetics. So this definitely facilitates the phenotype reconstruction. So we selected, together with our colleagues from the animal breeding department, we collected a list of 18 different regions and candidate genes. On the top, you see the MC1R gene, responsible for coat color. We also looked at a sister gene, MC4R, has nothing to do with coat color, but with fatness, growth rate, and feed intake. We also looked at a variety of others, but I'm only talk about results from these two genes. You've seen this slide before. We did a targeted enrichment, as Ben explained yesterday. And here are some results for a wild boar. That's from the north. He had the typical European haplotype, mtDNA haplotype, and we did both on the sample because it appeared to be a very well-preserved sample. So we did an enrichment for these 18 phenotypically relevant genes. It was quite good, 60%. In contrast, when we did a whole genome shotgun sequencing, we only got a coverage of 3%. As an example, for the MCIR1 gene, the coverage was so good that basically every base was covered, and an average of about 14 times. MC4R did not work so well, but also good enough to draw conclusions. So here you see an example. That is the enrichment we used for the MCIR1 gene. Coverage is very good, it's excellent. We could reliably call SNPs. This is, for comparison, this is the shotgun sequencing, and here really only a small part of the gene was covered. Now, the MCR1 gene has been widely studied in pigs, mainly by Fang et al. In 2009. They published this region joining network of the alleles or haplotypes, and they used domestic and wild animals from both Asia and Europe. All these alleles here, or haplotypes, these are alleles that are only, or that are found only in wild boar populations. And they are characterized, they differ from each other, these alleles haplotypes, but by synonymous mutation, as could be expected for a wild population. These are the camouflage colors, and other coat colors are not, or animals that have other coat colors are not expected to survive in the wild. These here are domestic coat colors, and these here as well. These are typical for the Asian animals, and these for Europe. And they differ from each other by non-synonymous mutations. They also put a line through here that is to separate the European alleles from the Asian ones. Now, when we looked at our wild boar number 5 with a very good coverage, we saw immediately, well, everything is fine. This animal carries the haplotype 101, which is the wild type, the camouflage coat color, and it's typical for European. So that was great. But in addition, we saw a change at codon 301. It's synonymous, and, but that wouldn't be a major issue, but surprise, surprise, this is a known mutation, but only in Asian wild boar, modern Asian wild boar. Okay, whoops, what's going on here? In addition, you also saw a similar, we made a similar observation in a domestic pig from the south. Here we also saw a change at a codon, also synonymous. Again, this we see only in modern Asian wild boar. So most likely, this animal or one of his ancestors picked it up from a wild boar, but how come that we see Asian alleles in a modern wild boar? According to this phylogeny, this is not supposed to be the case. These animals would cross somewhere in here. It's very difficult to put to consider this mutation because they were not part of this network at the beginning. Well, the easiest explanation to explain this discrepancy with the modern data is that Fang et al. Used a very small sample to determine the alleles. They had something like 15 wild boar specimens. And so, you know, if we analyze more, investigate more animals, we might also see so-called Asian alleles in European populations or vice versa. So that's a more likely explanation, but the more exciting explanation would be that maybe really 7,000 years ago in Northern Europe wild boar populations we had a larger diversity than today. So this needs further investigation. Now let's look at the MCIR1 variation in the early domesticates. And indeed, we found in one animal that also has the Y, the Near Eastern mitochondrial haplotype, we found that this animal is homozygous for haplotype or allele 501. And this is this haplotype here. It gives rise to a black spotted coat. The same we found for another animal, but genetically it's different. It's homozygous for a different allele 503, but it gives rise also to the same spotted appearance. So this was very exciting. We found also animals in southern Germany that are also that carry mutations, however, only in the heterozygous state. And we found something really neat in this animal from the south. We saw that this is homozygous for the haplotype 401, and 401 you find here, and this means that this animal had a red coat color. So based on the MCIR1 gene alone, we see already quite a diverse coat color appearance early on, black spotted and red. We also looked at the sister gene, MC4R, and we found something very interesting in this animal. You've seen this animal, data of this animal before. It's the one that has a Near Eastern haplotype. It has a black spotted coat color. And we found that this animal is heterozygous for a missense mutation, a very well-known missense mutation in modern breeds. And it's associated with greater fatness, faster growth rate, and feed intake. Even though it's only heterozygous, this mutation acts in a gene dosage-dependent manner. So it was also animals today that carry this mutation in a heterozygous state are fatter and heavier than the homozygous wild-type animals. So it seems that early on they went for the bacon, and energy-rich food might have been a selection criterion. Of course, we have to continue. These are only preliminary data, and we also sequenced the whole genomes, and we then will look more in detail how the wild and the domesticates differ from each other versus modern genomes. We will look also at signs of positive selection, cost of domestication, and of course the Y chromosome. And I'm particularly interested in also the mtDNA. Why was the Near Eastern haplotype replaced? Was it really only drift? Very likely, especially if we had really a small founder population. It's a likely scenario. Or maybe there was also selection involved. Maybe they didn't, these animals carrying the Near Eastern haplotype didn't thrive so well in the cold climate up in the north. Who knows? We will see. So what we can say already is that we've received in northern Germany what we call a Near Eastern starter kit of some domestic pigs but very soon they started their local breeding business, and it's a protracted and multi-stage process, and they incorporated to a large extent local animals. The early phenotypes that were selected for were coat color, and we see already quite diversity genetically, but also phenotypically, and it seems also fat deposition. And as we've seen with the discrepant data, the discrepancy between ancient and modern data, it shows again how helpful aDNA studies are to improve hypothesis based on modern data. And with this, I would like to end my talk and wish to thank my collaborators in the aDNA lab, the mathematicians who did the modeling, and our collaborators in we call them the animal people, animal breeder in the Department of Animal Breeding and Husbandry at our university, who all contributed to this study. Thank you very much. Okay, not too bad.
Thank you, Almut, very much. So, any questions? Yeah?
I'm interested in your targeted enrichment. So, if you were to generalize, would you say a 3% bone is one that will work well for enrichment, or could you give us some guidelines in that? Or
we tested many samples, something like, well, not many, but sort of at least 10 or 12, and this wild boar was the best of all. And we can't really say which one will work or will not. And but the targeted enrichment for sure is for us here at the moment the way to go, and we'll do the genome sequencing only for those samples that show already very good performance in the targeted enrichment. So we select.
Is there any particular reason why MCAR worked less than MC1?
Worked less?
Yeah.
No, I don't know. No.
You don't know?
No.
But it does work less. Right?
It seems, yeah. Overall animals, it seemed to work less. Yes, that's true.
I'm just curious, what's the hypothesis about the selection on skin color for pigs? So what's the advantage of having lighter skin and darker skin in the environment?
Yes.
I mean, always when I see them in the summer, they have sunburns.
No, this is a very good question. Archaeologists have wondered about it for a long time, and maybe it was easier for them to keep the animals, so to spot them. It may sound a bit trivial, but why not? It facilitates the handling of the animals if you can spot them, and or maybe they just liked the exotic appearance of them. You know, instead of having a boring gray coat color, maybe they went for the red. You know, I don't know.
Consider looking or investigating additional coat color genes like
Sure, as a kit and so on. Yeah, ongoing. It's still very preliminary data. right? Of course.
If there's no other question, thank you very much.
You're welcome.
Proceed to the next
Yeah.
presentation.